In 1891, surgeon William Coley began one of the most bizarre experiments. He injected mixtures of Streptococcus pyogenes and Serratia marcescens bacteria into the tumors of cancer patients. It seems crazy to infect cancer patients with bacteria, but with this he achieved over 1,000 complete and lasting tumor remissions in various types of cancer. In other words, Coley cured cancer with bacteria, and today we can replicate his success, but without using bacteria. Without infecting anyone, without causing diseases. In fact, 2 Brazilians have already been cured by a modern version of Coley's idea to treat cancer. But even with this success, the idea of injecting bacteria into tumors was not taken very seriously by the scientific community at the time. And there are a few reasons for this. At the time, it wasn't well understood exactly how bacteria cured cancer, and the other is that nobody really liked Coley. But the fact is that this was the extraordinary beginning of new technologies that today can help us answer the question of how close we are to curing all types of cancer. So stay with me until the end, because this video is going to be fascinating.
Hey, Pedro here. This video you are watching was originally in Portuguese, my native language. This is the attempt of our team to translate it to English, and I sincerely hope you enjoy it. Your feedback is extremely important to us. Now, back to the video.
In 1957, Lewis Thomas and Frank Brunet found a possible explanation for the strange success of the treatment developed by Coley in the so-called tumor immunosurveillance hypothesis. The two researchers noted that the tumor immunosurveillance hypothesis observed that tumors grow and multiply rapidly in animals and humans with compromised immunity, which confirms the importance of the immune system in suppressing cancer. And better understanding how this cancer immunosurveillance effect works could be the key to advancing treatments for all types of cancer. But let's take a step back. What exactly is cancer immunosurveillance? Cancer immunosurveillance implies that the immune system will recognize and destroy normal cells before they transform into tumors. It will also be able to recognize and destroy tumor cells in already formed tumors. For example, when William Coley injected his patients with bacteria, the injection stimulated the immune system, which was activated to destroy the bacteria, including those in the tumors. And once in the tumors, the immune system cells were also able to destroy cancer cells. A very important cell for cancer immunosurveillance is the T lymphocyte, particularly the type known as cytotoxic. Cytotoxic cells are capable of destroying tumor cells by releasing toxic substances into these cells. And not just tumor cells, actually. Cytotoxic cells are also capable of destroying various types of cells. But they don't work on their own. They need a trigger to start destroying other cells. And the main stimulus is the recognition of antigens, which are the small protein fragments produced by tumor cells. When the immune system recognizes tumor antigens, the lymphocytes are activated to search for cells that have these antigens. The T lymphocytes travel throughout the body, tracking cells with the antigen. This is like the cellular version of checking someone's documents. When our cells present some strange proteins, the immune system acts to prevent a tumor from developing. A cell that is producing a strange protein might be a tumor cell, and the immune system takes this threat seriously. But this raises a problem. If our immune system does all this, why do we still have cancer? Cancer cells have their own strategies to escape immune system responses. Tumor cells can prevent the immune system from accessing their antigens, and thus it cannot recognize the threat. It's the cellular version of the excuse "I left my wallet at home" to avoid having to show identification. Additionally, tumor cells can produce immunosuppressive substances to decrease the efficiency of the immune system. The good news is that there are various treatment possibilities to suppress the mechanisms tumors use to escape the immune response. To be direct, it may be that the cure for cancer consists of improving the immune system's ability to deal with cancer, but it's not that simple. Even cancers that affect the same organ do not express the same antigen molecules. For example, in melanoma, which is a type of skin cancer, there can be 1,000 different mutations per cell of the same type of cancer. Tumor cells form from DNA mutations that arise due to uncorrected damage in the genetic code of our cells. The damage that causes mutations has various sources, from exposure to radiation to habits like smoking and alcohol consumption, or even genetic predisposition. The classes of genes that are most affected by mutations are those involved in cell growth, genes associated with tumor suppression, genes that regulate cell death, and also genes that repair damaged DNA. These are precisely the genes that prevent the onset of cancer. But when they are damaged, the development and growth of tumors is favored. The human genetic code has more than 3 billion base pairs, with over 20,000 genes that can undergo mutations. There are many possibilities for mutations in the genetic code, and these mutations may or may not impair the function of these genes. Most mutations will not lead to the development of cancer, But since the genetic code itself is diverse, this implies a diversity of possible types of cancer with different behaviors. And it is precisely because of this diversity of tumor cell types that traditional therapies, like chemotherapy and radiotherapy, are aggressive to the point of destroying normal cells and causing undesirable side effects. It is difficult to make a treatment target only cancer cells precisely because of the diversity of protein expression in these cells. Cancer is a disease that manifests in very different ways, which is why it is so difficult to produce a single cure for all types. But difficult does not mean impossible. With knowledge of immunology, we are able to obtain more specific treatments with better results, such as treatments based on monoclonal antibodies. Antibodies function to connect to specific molecules within our body in order to then cause a designated effect. One of the effects is to signal the cells of our immune system to the presence of an infection or a cancerous cell, and monoclonals. Well, I would need a whole video just about the immune system or infections to properly explain monoclonal antibodies. If you want a video like that, type a muscle emoji in the comments. And today, thanks to knowledge of molecular biology and biotechnology, we know that we are capable of creating antibodies that bind to any molecule, including molecules involved in the proliferation and growth of tumor cells, which prevents the tumor from using these molecules to proliferate. Other antibodies block growth factors important for stimulating the formation of blood vessels, which will nourish the tumors. Thus, the tumor cells are left with little supply of oxygen and nutrients. Even more impressive is that there are monoclonal antibodies that bind to tumor cells to deliver a drug capable of destroying the cancerous cell, like chemotherapy drugs. In this way, chemotherapy affects cancer cells without causing as much damage to the rest of the body, and the uses of monoclonal antibodies don't stop here. We can also use these antibodies to bind to the tumor cell and make it the target of the body's own immune response, or prevent the cancer cell from using its strategies to avoid being destroyed by the immune system. In this way, immune system cells can destroy the tumor cell by releasing toxic substances or even engulfing it, similar to what happened in patients infected by William Cole but without needing the infection. But despite being successful, monoclonal antibody therapies have a very high cost. And it is still necessary to research how to make the treatment more practical and accessible. Monoclonal antibodies are extremely promising in treating various types of cancer. But we can go even further. You can use your own immune system to destroy tumor cells. I told you this video was going to be fascinating. This is CAR T-cell therapy, which in 2019 cured the terminal cancer of Vamberto Luis de Castro. Vamberto suffered from a very aggressive non-Hodgkin lymphoma, and even with the disease already advanced in the patient, the CAR T-based treatment reduced his symptoms in a few days, as well as drastically reducing the number of cancer cells in just 30 days. In 2023, Paolo Peregrino had been suffering from cancer for more than 13 years. And with CAR T-cells, he had complete remission of the lymphoma in 30 days. It's even hard to explain how incredible this is. A few years ago, these news belonged to the realm of fiction. So how did CAR T-cell immunotherapy achieve this feat? The treatment begins with the collection of T lymphocytes from the patient's own blood. These lymphocytes are prepared in vitro to carry a specific recognition molecule for the patient's tumor cells, called CAR, which stands for chimeric antigen receptor. The CAR is a mixture of two types of molecules. One part of them serves to recognize the tumor, and the other part to activate the lymphocytes capable of destroying the tumor. Then, the patient receives these lymphocytes back, but now they will be able to recognize and destroy the tumor cells. So far, this treatment with modified T lymphocytes has only been used for hematological cancers, blood cells, and we still do not have its use in solid tumors. The big challenge is to identify tumor molecules that are only in tumor cells and not in normal cells. After all, we don't want normal cells to die. Although CAR T cells are very promising, we also have other personalized therapies that are being studied. For example, the idea of cancer vaccines, which try to teach your immune system to recognize and fight tumor cells in a way very similar to how traditional vaccines teach your body to recognize infections. The difference is that cancer vaccines are a treatment, not a way to prevent and mitigate the disease, as in the case of vaccines for infectious diseases. Even though they are promising treatments, it is important to make clear that immunotherapy is also subject to various limitations. The body may react poorly and trigger a strong inflammatory response. Or sometimes immunotherapy can cause tumor cells to adapt to escape the immune response. But even though cancer is still seen as a serious disease, with complicated treatment and generally a poor prognosis, this is changing. With the evolution of cancer treatment, the possibilities of cure are greater, because most cancers are treatable and many of them can be cured. Until recently, a melanoma diagnosis represented a death sentence. But with immunotherapy, scientists are able to cure 60% of patients with melanoma. But it's not just immunotherapy that cures. There are many patients who have had their cancer cured with radio and chemotherapy. And these therapies do not need to be abandoned because of new ways to treat cancer. Radio, chemo, and immunotherapy will continue to be tools against cancer, and these therapies can be combined to achieve the best possible results. A new therapy represents a new way of dealing with the disease using science, research, and human effort. And with immunotherapy, William Cawley's idea of using the immune system itself against cancer opens a new window for possible treatments. Unfortunately, we still don't have a cure for all types of cancer, but new research indicates that it may indeed be possible to cure every type of cancer. Or as the immunologist Jim Ellison, who was the 2018 Nobel Prize in Medicine, says, "We don't have a cure for all types of cancer yet, but one day we might." Thank you very much and see you next time.